So the cloning of Dolly, it was really an inspiration for me and for all of us here. So it's a great, I'm really thankful to the organisers for giving me the opportunity to talk to you today. And I'm interested in differentiation and in understanding how pluripotent cells choose their fate. And of course, we know that in the embryo, they choose their fate according to their position with respect to signalling centres. And so accordingly, we can direct their fate in a dish by giving them the right signals. However, we and many other labs frequently observe that even uniform monolayers of pluripotent cells seem to adopt different fates for reasons we don't understand. So one way of thinking about this could be to take into account that, of course, most other cell types don't just passively receive signals, they actively modulate their responsiveness over time. And so if this is also true of pluripotent cells, then perhaps we could use this as a way of asking, does signal responsiveness predict the future behaviour of cells? And if this is true, we could use this as a way of going in and interrogating the other things which are influencing differentiation. Now, in asking this, we're focusing on two kind of differentiation decisions. First, this transition from naive pre-implantation to primed post-implantation pluripotency, and from there to lineage choice. And these can be modelled very nicely in culture using conditions which in part were developed by Paul Tesser, who's here today. And so if we wanted to, we wanted to follow how cells are changing growth factor responsiveness during these transitions. And we decided to start by looking at BMP, because it regulates these transitions in vivo and in vitro. But so do other signals such as FGF and nodal. But with BMP, we have a particular advantage, which is that we know of a single direct target gene, ID1, which can recapitulate these effects of BMP. And we can easily detect ID1 protein. We have good antibodies. We have fluorescent tagging strategies. And so essentially, we have a biologically relevant readout of this pathway at the protein level. And so I'm lucky to have this excellent postdoc in the lab, Matthias Malaguti, and I'm just going to show you a lot of Matt's work in one quick slide, which is to illustrate that BMP responsiveness does indeed change dynamically during early development, and there's a clear peak of responsiveness, as read out by ID1, at this peri-implantation stage of development, or the culture equivalent. And this is exactly the time that cells are transiting between these naive and primed states. So they're just starting to lose nanog. And Matt noticed that ID1 is exclusively found within these cells which have recently lost nanog, but only in a subset of these cells. So this was very interesting to us because we already knew from Ian Chambers' work that if you lose nanog, it doesn't commit you to differentiate into this primed state, but rather there's always a subset of cells, although we couldn't predict which ones, that would resist differentiation and go back. And so we wondered in this respect whether our ID1 cells were special in some way. So to test this, Matt made this dual reporter line so we could get rid of the nanog high cells and focus only on this interesting nanog low compartment and ask, within that compartment, is there a difference between ID1 high and low cells? And at the transcriptome level, the answer is no. They look almost identical. So it was therefore very surprising that they had rather different behaviours when we challenged them with this few hours of differentiation conditions. So essentially, the ID1 high cells, or BMP responsive cells, behave more like nanog high cells in that they tend to resist differentiation, where it's the ID1 low cells that are more susceptible for differentiation. And so having seen this difference in behaviour, Matt went back to the transcriptome data, looked at this small handful of genes that was differentially expressed, and saw at the top of the list were these two genes, which are direct readouts of FGF and nodal signalling, respectively, which is very interesting because these, as Paul Tesser has shown, are the two signalling pathways that sustain this primed pluripotent stage that the cells are moving towards. And so what we think we're seeing here is by looking at ID1, we're seeing a coordinated change in signalling and differentiation. Is ID1 responsible for these changes? We think yes, essentially because ID1 low null cells behave like ID1 low cells. They have a higher chance of differentiation. They have higher FGF activity, no difference in nodal, which we have ideas about if you're interested. And we can suppress this phenotype by forcing the cells to remain in this nanog high compartment. So this is what we think is happening. And the reason we think this might be important is because if you can think about these naive cells, they've got nanog and all its friends to protect pluripotency. And we know in this state, FGF drives cells out of pluripotency into extraembryonic lineages. So when they lose nanog, they have to take this rather precarious journey towards this prime state. And the reason it's precarious is because if they acquire FGF activity too early, they'll lose pluripotency. And so we think ID1 kicks in just at this kind of fragile transition point to coordinate signaling with differentiation. Now, okay, so what we decided to do then was to follow this up by looking further downstream of ID1 to see if we can find anything else interesting about this transition state. We know ID1's not a transcription factor, it doesn't bind DNA, it only binds other proteins. A few years ago, we published a yeast-2 hybrid screen where we identified this factor, TCF15, as being suppressed by ID1, and it also has these other interesting properties, which led Chai-Yi Lin in the lab to suggest that maybe this factor, TCF15, perhaps now identifies those cells which are now more receptive to differentiation cues. And indeed, this turns out to be the case. So it's the TCF15 high cells which respond beautifully to differentiation conditions, in this case, generating lots of neurons in this assay, and it's the TCF15 low cells which seem to be resistant to differentiation cues. And but what was more interesting really, was that they behaved identically if you tested them under self-renewal conditions. So this means that they're not yet committed to differentiation. So Chai-Yi was very happy about this because she realized what it meant is that she's now found a marker that allows us to identify this state before commitment, but which predicts the way cells are going to behave in the future, and then ask what it is that's special about these cells, what's different about them. And one thing that she noticed straight away was different about them was the morphology of the colonies they form. So the TCF15 high cells form flatter, more epithelial colonies, even though they're not yet epistem cells. And we're in the process of using new image analysis tools, which I'm about to describe in a moment, to really explore how TCF15 is regulating this difference. But what I really want to talk about today, in the last bit of the talk, is why we were interested in this morphological difference in the first place. And this was because we know in the embryo that these two transitions are accompanied by these morphological events, the formation and disruption of an epithelium. But we'd kind of assumed before that these were just a consequence of the differentiation events. But Chai-Yi's data started to hint that maybe these morphological changes could be happening before differentiation, raising the possibility they could be influencing the differentiation process. So we wanted to explore these links a bit further. And here we had a problem because at the time there were really no tools available that allowed us to measure the way that cells are organized in 3D space and correlate this with differentiation events. So fortunately, we had this really multi-talented postdoc in the lab, Guillaume Blanc, who, as well as being trained as a biologist, has also, over the last few years, written some new image analysis software that we call Pixels, which has a number of important features. I'm going to explain a few of those to you now. So first of all, Guillaume's managed to solve the challenge of accurately identifying individual nuclei, even within very complex, crowded tissues, such as this mouse embryo. The software can also identify all the individual centrosomes, and it can solve the rather difficult computational challenge of associating each centrosome with its own nucleus. And to explain why we want to do that, I'm going to use cakes, as you do. So you can imagine that if the computer can see every nucleus and its centrosome, it can draw an arrow between them to give every cell a vector, an orientation. And then by looking at how these arrows are organized relative to each other, we can, for example, detect the emergence of an epithelium, as we see here. Now, Unfortunately, as you all know, real life isn't always as simple as cakes. So we're not just looking at the orientation of these cells in a nice two-dimensional plane. We're looking at their orientation in 3 dimensions. And we're not normalizing them to a nice straight edge of a computer screen, but rather, in this case, we're normalizing them to a complex curved surface of this gastrulating embryo. So Guillaume has also devised methods of automatically detecting complex 3D landmarks within embryos or tissues, such as surfaces and cavities. And then we can normalize the vectors of all the cells to these landmarks. So in this way, you can start to get the idea of how we can start to correlate the organization of cells with differentiation. But I think what's really important about this approach is that we don't just see the general trend of how cells are organized in a particular region, but we can also go in at a single cell level and identify cells which are starting to deviate from that trend. Okay, so this becomes particularly important if we return to our cell culture system. So this is the picture I showed you at the beginning of cells undergoing this apparently unpredictable differentiation. And just very briefly, what Guillaume has been able to do now is he's been able to detect individual cells within this epithelial sheet, which are starting to diverge from the epithelial structure. And he showed that this happens before the cells commit to differentiation, but that it correlates with an increased probability of them following neural or mesodermal fates in the future. And this is based on particular markers of these states that were described by Anestis Tsacharidis and Val Wilson in a nice collaboration with Guillaume. Now, so now Guillaume is very happy because this has been a completely independent line of evidence that's converged on Chai's work to start to suggest that there are changes in cell orientation that might be predicting the future behavior of the cells. So this raises the very interesting question of whether they have any influence on differentiation. So just in the last couple of minutes, couple of moments, I'm just going to start to show you a little bit about the approaches that we're taking to address this rather challenging question. So in this example, what Guillaume has done is he's essentially manipulating the matrix that the cells are exposed to. And he's shown here that this is able to randomize this nucleus centrosome vectors with what would otherwise be an epithelium. And the consequence of this seems to be an increase in differentiation. Now, importantly, we think that this differentiation cannot simply be attributed to the exposure to the matrix, because if we expose the cells to the same matrix in a different context here, where we're actually reinforcing rather than disrupting the epithelial organization, in this case we see a suppression of differentiation. So we think that we observe that the differentiation events always correlate with the changes in orientation and not just with the exposure to matrix. And then finally, just as an alternative approach, Guillaume's been using micropatterns. So here, by physically constraining the cells, again, he's able to randomize the nucleus centrosome vectors and again see an increase in differentiation. And the reason that we like this approach is that we can use all these different shapes of micropatterns to try and control for all the other events which would usually accompany morphological changes and which might also be influencing differentiation, such as physical forces or the development of signaling gradients. So in this way, we're hoping to uncouple all these influences, and we think we're starting to reveal a specific role for the orientation of the cell in influencing their future differentiation behavior. So I'll stop there and just summarize what I've told you. So I told you there does seem to be a coordinated change in signaling prior to differentiation, and this seems to be coordinated by ID1. I didn't have time to talk about the other interesting side of this question, which is what controls, what influences these changes in ID1 and generates these differences in the first place. And then by following downstream from that finding, we're starting to get evidence that changes in cell orientation may be influencing the differentiation of the cells. And again, I didn't have time to explain why we think that might be important in the embryo. And then finally, I've introduced some features of this new tool, PICcells, which we really hope is going to be useful to anyone in the community who's interested in looking at tissue organization and cell differentiation. So I'd just like to finish by thanking everyone in my lab and all our collaborators who've contributed to this work and to our funders. And thank you for your attention.
Okay, we have some questions for Sally. Yes.
Beautiful talk. Thank you so much. This was really interesting, in particular, these two complementary approaches, and that was exactly my question. So you have now molecular markers like your TCF15, I believe, and ID1. On the other hand, your morphological changes that you see in the cultures. Is there any correlation between the two, or are they the same populations?
Yes. So we think, although I didn't really show any data on this. So we think that TCF15 might be one of the things that's actually controlling this change in morphology. So we think that's the first event, is this activation of TCF15, and then the cells start to change their orientation downstream of that. So we're kind of trying to link things up in that way, if that addresses your question. But ultimately, the way that we think about it is that, what we'd really like to do is look at the effects of transcription, signaling, and morphology, which are all feeding into each other, right, in this kind of positive feedback loop. And what we really hope to do with this quantitative imaging is to look at the relative influence of these things over time and sort of put them into a kind of one network. That's the ultimate aim.
Right. That was sort of my question. Is you think it's first a chromatin switch, so to say, that is the initiator of this decision, or is it a morphological change?
So this we don't know. So we've not looked at all at the chromatin changes, but we have the markers now to be able to do that. So this is what we must do next. So it's something we've not looked at all yet. It's very important.
When you talk about cell orientation and you indicated physical forces driving this cell orientation, actually in the picture that you show us, there seems to be multiple vectors. Yes. What are the physical forces that are you thinking about?
So there are several papers that suggest that in pluripotent cells and in other cells, when they're subject to so for example, when they're at the edge of a colony and subject to more strain, this somehow influences their upregulation of mesoderm markers, for example. So the mechanisms aren't well understood, other than perhaps hippo signaling might be involved. But it's these kind of studies that we're thinking about. And we're thinking about how we can untangle those effects from the polarity effects. Okay.
Thank you.
So thanks very much for the great talk. The question is, the changes in orientation seem to precede and predict general differentiation, but I'm wondering more specifically if you can dig further in whether specific changes in orientation might predict mesoderm, endoderm, ectoderm.
So that's exactly what we want to do. So at the moment, we're focusing the germ layer side of the project on mesoderm, but we have another project I didn't have time to talk about because it's already published, where we show that if you manipulate epithelial cadherin, this really influences neural differentiation as well. And we don't know yet whether that's part of the same thing we're seeing with the cell orientation or it's a different mechanism. So that's the kind of thing we're going to try and look at in the future.
Okay, thanks so much, Sally. Let's move on.